Hello, everyone, and welcome to today's webinar, Single-Cell Spatial Phenotyping, Fueling the Next Wave of Discovery Biology. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Akoya Biosciences. To learn more, please visit akoyabio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have trouble seeing or maybe hearing the presentation. I would now like to welcome our speakers, Dr. Jonah Cool, Program Officer, Single Cell Biology, Chan Zuckerberg Initiative, and Dr. Oliver Braubach, Head of Research Applications at Akoya Biosciences. For full biographies on our speakers, please click on the Biography tab at the top right of your screen. Welcome, Jonah. You may now begin your presentation.
Great. Thank you very much for the introduction, and also to the LabRoots and Akoya team for putting on this webinar. It's really a pleasure to be here, and I'm excited to very briefly share some of our work at the Chan Zuckerberg Initiative, and specifically the work of the team that I lead focused on single-cell biology. It will be a brief overview, and so just to reiterate the moderator's encouragement to ask any questions or please engage. Really looking forward to the discussion and hopefully digging into some of these areas in greater detail. So let me start with an overview in the mission statement of the Chan Zuckerberg Initiative Science Initiative. And again, we have a number of other initiatives and would encourage people to go to our website and learn more about those, as well as some of the programs within the Science Initiative that I won't have the time to talk about. So our mission is to support science and technology that will make it possible to cure, prevent, or manage all disease by the end of the century. And this is obviously a very, very lofty and ambitious statement. And there's two things that I would like to highlight that give us hope and encouragement and feel convicted that this is indeed a mission that we can accomplish as a group. So the first is the support of science and technology. And my presumption here is that the interest in this webinar is that there are these new, exciting, and foundational technologies that are opening the door and shedding light on important fundamental aspects of science that are giving us an ability to understand healthy and also specific diseases in new ways and through new light. And that by working on these technologies and deploying these technologies, advancing them and sharing them with communities openly, there's the possibility to make broad-scale changes that will advance science and medicine in a non-linear way, so exponentially. And the end of the century is still a long way away. And so there's a lot of possibilities and opportunities for this acceleration of progress by making initial work and focus on science and technology. And so it's a statement both about the importance of fundamental discovery and research, the role that technology has to play in that discovery and research, and also the ability of us to make unprecedented advances over the next 80 or so years. We have a number of active programs within the Chan Zuckerberg Initiative Science team. These include very technology-focused efforts, such as our work in open science and imaging, as well as more disease-focused areas such as neurodegeneration. And again, would encourage people to go onto the website and learn more about that. What I'm going to speak specifically about is the work within the single-cell biology program. And within this group, our goal is to accelerate and support single-cell biology methods, technologies, and applications. And the important thing here is that level of resolution of the single cell. And that is really what we're focused on. So there are a number of methods that have emerged and are rapidly scaling, as well as countless other opportunities to refine and develop new methods that provide insight into diverse molecular features, the spatial organization of cells within intact tissues that are really what we're interested in supporting and focusing on. And so the level of resolution here is the thing that is the common thread through the work that we are supporting, as well as some of the tools that we are actively building at the Chan Zuckerberg Initiative to hopefully help support you and your work, as well as others in the field. So let me tell you briefly about the current opportunities and funding initiatives, and also some of the tools that we are building. So the Chan Zuckerberg Initiative is a unique philanthropy where we are both funding external scientists, such as many of you that have joined today, as well as building open source tools that we hope will support your work. So these are freely available and openly available and are really intended to be used by the community. And so we are both grateful and hopeful that they will solve some problems and challenges that you face, but also eager to hear your feedback and understand how they might be able to be evolved. So the first two areas here are active areas of funding within the single cell biology program. The first is support for a large international community that is looking to build a reference map of all the healthy cell types in the human body. And this is the Human Cell Atlas. And I think and hope that many of you have heard about this. It's supported by many funders around the world and would encourage you to go to the Human Cell Atlas website and you can register and join that community for additional updates. And so here we are supporting data generation efforts. We have supported computational methods and analytical developments and benchmarking, and are really looking to support the data generation, the analysis, and the sharing of the human healthy reference data sets. In addition to the reference data, we are also supporting some smaller initial programs and pilots focused on single cell analysis of inflammation. And so here is an opportunity to use that reference data, to use the tools that are being developed to support and generate that reference data set, but to begin to see how that informs and changes our view of complex biological processes and diseases such as inflammation across various tissues and diseases, with the goal of understanding them at the single cell level and ideally identifying pathways and programs and cellular interactions that are perhaps common and might inform our view of shared mechanisms or prioritized targets. And then, as I mentioned, in addition to those funding initiatives, we are also actively supporting and building tools. And for the rest of the talk, I'll focus on one of these, which is a data exploration tool called Cell by Gene, but also wanted to briefly highlight another tool that we have developed and is available to the community called Starfish, that is a analysis tool for image-based transcriptomics or other image-based assays, including potentially Codex, it's been used in the past. And then also our support of the Human Cell Atlas data coordination platform, that is an effort to build a cloud-based infrastructure to store and serve the data being generated by labs that I mentioned at the top. And again, we'll briefly mention that all of the groups that we have supported for these first two areas, as well as all of the resources that they've generated, whether it be code or protocols or preprints or data, are available and listed on our website, and we make an effort to keep that up to date. So please check that out for more. So let me now transition to an example of one of those technology tools that we have developed and are actively developing. It's called Cell by Gene. And the fundamental goal of Cell by Gene is to develop and make accessible a scalable collaborative analysis tool. And so this hopefully reduces the friction and enables teams of researchers that are looking at high dimensional and complex single cell data sets to more easily collaborate and understand and gain insight into their data. It is also a hosted data publishing and exploration platform, so that when groups are ready to share data, and especially as the demands on that data grow with growing data set size or complexity, that there is a hosted solution that is free and available to researchers and makes it easy for them to host, takes away the maintenance burden of some of those portals, and similarly makes it easy to find data and explore data if you're looking to understand more about a given tissue or cell type or different questions that would fuel your hypothesis. So what Cell by Gene looks like in this workflow is we have some support and scripts for validating a common schema that's really meant to provide interoperability among these data sets, such that if there's an interest in reuse of the data or integration of multiple data sets, that hopefully the bar to that is lowered and you are enabled to do it. And so you can share your data and validate it in this common schema. We'd be happy to talk a little bit about that and also answer questions offline if you're interested in contributing data. And then it allows you to publish into these collections, which are thematically linked. It could be a paper. It also could be a larger body of work that is maybe associated with a given lab or working group or anything like this. Once the data is shared publicly, it then allows users to either in the browser launch a exploration tool. And again, this is very scalable. So we currently have data sets up to 4 million cells that can be viewed, and differential expression can be computed, et cetera, on different subsets of the cells. But it can also be downloaded, and the download can be done in a number of different languages. So you get the processed matrices in that unified format in the format of your choice, hopefully to make it very easy to pull it into whatever downstream tool chain you're looking at, such as Seurat or ScanPy or Bioconductor. So this is what the collections look like. Again, you can go to the Cell by Gene website and check out what is currently available. As I mentioned, it can have multiple different data sets associated with a given collection. It can also have different types of data. So I want to highlight here, there's an example of a transcriptomic data, a proteogenomic data. So certainly protein information is something that we're really eager to adopt and work on. And again, very excited to think about the possibilities of working with more Akoya and Codex data, as well as other data types. So please check this out and let us know if you have any questions. At the moment, the data resource that's available to anyone or any of you that are interested is about 36 collections. This is probably an underestimate, actually. The number continues to grow. Within those collections, there's over 150 data sets and now nearly 20 million cells worth of data that are available. And also want to highlight, this is not just single cell transcriptomics. It is very diverse. So 7 different assay types, including functional assays like PatchSeq, and as well as dissociated and spatial assays. This is the email if you would like to reach out about contributing data or have questions about any of the possibilities. So finally, I wanted to emphasize that the flexibility of the tool. So it really will allow scalable exploration of any matrix. And so that certainly includes a matrix that includes spatial coordinates. So here are 2 quick examples of data that is currently available. One was generated by the barcoding of arrays and spatial transcriptomics in a beautiful paper published earlier this year from a group in Oxford and Alison Simmons, looking at fetal development of the gut. And another example of image-based transcriptomics, and there's a few of these. So this is an awesome fish data set that was published by Sten Linnerson's lab in Sweden, but also there is data available from methods such as MERFISH and SeqFish and other methods. Would like to make the point here that this is all transcriptomics, but again, there's nothing to say that we can't work with protein data, and this is something that we're actively pursuing and would be available. So if you have those data sets and would like to share them or explore them with Cell by Gene, please reach out. We'd really like to support you and would be excited to have those available to the community. So with that, I will end. I will thank everyone. These are a couple different links and ways to get in touch and stay abreast of the research and work within CZI and CZI Science, as well as my personal Twitter handle. We'll also mention that our team is hiring if any of this work was inspiring and you would be excited to work on it. Looking forward to the Q&A, but before that, I am excited and eager to introduce Oliver, who will take on the next section. So with that, I'll hand it over to you, Oliver.
Thank you, Jonah. Thanks for that intro, and thanks for the nice information on the cool stuff that you guys are doing. I met Jonah, obviously, through this CZI work, and I'm very curious to see what our data will look like on this platform as well. So I'm the head of research applications at Akoya Bioscience, and I must apologize in advance. I went to a karaoke bar last night to celebrate the almost return back to normalcy. So I might have a sip of water here and then, see if my voice holds. But what I would like to talk to you about today is sort of a follow-on to this idea of single-cell biology. Evidently, that's what we all aspire our research to get to today, to generate an atlas of the entire human body of every cell inside of it. And we're really, Akoya is a spatial biology company, so we have this concept that we call single-cell spatial phenotyping. I'm going to explain to you how we see that based on the Codex experiment, the Codex technology today. I'm going to walk you through an experiment first, and then I'm going to talk to you about how this Codex system works. I'm going to give you some views on why it really matters that you have single cell resolution in your experiment, evidently, because we're doing single cell biology. So single cell spatial phenotyping is a concept essentially where you're doing phenotyping on a whole tissue, and you have unrestricted access to your tissue, meaning that you can see every cell. Now, a couple of things that you need for that. First of all, you need obviously a nice piece of tissue. What you can see here is a piece of breast cancer tissue. It's rather large. It's about 2 millimeters from bottom to top, 3.6 millimeters from left to right. And this particular image shows you that breast tissue stained with 4 different markers, one of them being a nuclear stained DAPI. And the reason I always show this one up front is because it's really one of my favorite snapshots that just shows you beautiful resolution. So you can see these cells here. In blue, we have the DAPI nucleus. If you look somewhere around the center, some of these DAPI nuclei have a red ring around it. That's your nuclear envelope. And then outside of that, there's a cyan ring around that. That's your plasma membrane. So not only do you have single cell resolution, you actually have, to some degree, subcellular resolution. And when you're studying single cells, this might be perfectly obvious to you, you need to see them. You need to see every single cell on your image. And by using optical microscopy for our technology, in this case, this is a Codex experiment that we imaged with an epifluorescence microscope. That's a standard workflow. We ensure that every cell is seen. So that's a very important part, very important parameter that we need for single cell spatial phenotyping. Next, of course, is you have to be able to label your tissue with ample biomarkers. So you can see the same breast tissue now, and the program is cycling through different frames right now. Each of these frames shows you that same tissue labeled with 5 or 7 different antibody combinations. Each one of these images tells its own story. Now, I remember 10 years ago when I was a graduate student, I spent, months and months analyzing, 3-plex images. Most of my publications from back in the day were, 3 colors, 2 antibodies, 3 antibodies, at the most 4. But today we work with up to 40 antibodies, 42 antibodies, even more. So you just imagine how rich these data are. So you've just gone through that in this breast tissue. This is an experiment where we did 36 biomarkers in a single tissue. So how do you register all this information? What do you do with all this information? How do you get to this single cell spatial phenotyping result? So what you can see here is the same tissue has now been segmented out into every single cell. Essentially has been registered, has been segmented, and it also has a color code. In this case, this color code relates to the phenotype that we've identified. So the way we read our data out, the data structure that you get from Codex experiment is like this. You can see this table here in the middle. The first column is your cell. This particular tissue slab that you see has 63,000 cells on it. Each cell is then labeled with a combination of 36 antibodies, AB1, AB2, and so on, until you get to 36. Each antibody, or each cell, then has a combinatorial labeling pattern, which makes it uniquely identifiable. These data are very similar to what you would see coming out of a flow cytometer or a single-cell RNA sequence experiment. You have a lot of cells, a lot of different biomarker patterns. What makes the Codex data richer and different is obviously that you also have XY coordinates. You know exactly where every cell is, and that really is the gist of this. We know 63,000 cells are here. They're labeled very differently, and they're located precisely here and here and there. So that is really, really, really important part, obviously, of spatial biology. So if we go next and see how we essentially go and analyze these data, what we do essentially, just like you would in an RNA-seq experiment, we cluster our data. So you can see the output of the clustering in this round white area on the bottom left of the screen. That's a t-SNE plot. This t-SNE plot has different shades of green, yellow, blue, and red. So these shades show you different cell phenotypes, obviously. Each dot on the t-SNE plot is a single cell. Now, it's important to know that every one of these dots, every single cell, remember, is a cell that's on this morphological image that's here in the background. That means I can see the same cell on the t-SNE plot that I can see on my tissue image. That's immensely powerful, because oftentimes when you work with a technology where you don't actually see your cell, say, for example, you do single-cell RNA-seq or so, we know they're there. We know they passed through your sequencer. We know something's there, but it's always really a nice thing to be able to go back to the tissue and say, oh, there's my cell. And here's a really cool example of this. So after we clustered these results, we found 546 basal cells. You can see that on the right-hand side here in the text. Out of 63,000 cells, there were 546 cells that we identified as basal cells. That's less than 1% of the entire population. 44 cells were identified as a type of luminal cell. So this is a type of epithelium in the breast tissue. This is a very, very rare cell type that was first, I think, described by Kai Kessenbrock at UCI. Kai is actually a CZI researcher, so these data are consistent with what he found. And this is a tiny little proportion of the entire picture. 0.07% of the entire population of cells is this type of luminal cell. So if you jump back over onto the t-SNE plot, the very left-hand side of the t-SNE plot, you can see this red little island, and from it is this little tiny cluster of orange cells. So when I was first analyzing these data, I thought this was noise. I didn't know what to make of this. I had no preconceived idea of this tissue in the first place because this is not my expertise. However, it didn't really matter what we tried to do. This result always presented itself, these orange little dots here. 44 cells, who cares? This must be noise. But then think about it and go, look up at this little white circle here. See those 3 little orange cells right up there? Those are actually those cells. So having the visual of your cells, seeing them, not only on your clustered data, not only on a heat map, but actually on your tissue, is probably one of the best validations of your experimental data. The cells are there. We can see them. This is not noise. It's just an incredibly rare cell type. So that is really the power and the beauty of this single-cell spatial phenotyping business. You can look at thousands of cells, 500,000 cells, 1 million cells in a day, and you can even find the rarest cells there are. So this big piece of breast cancer tissue here, you can see all of these little orange dots. They sum up to about 44 of these cells. Again, this is less than 1 percentage point of the entire cellular population. That was identified in a completely unbiased way. I would actually say accidentally using this Codex experiment. I'm a neuroscientist. I studied breast cancer tissue because I needed a substrate to test my system, and I essentially stumbled across these cells. Now, once again, they've been described before in humans by Kai Kessenbrock's lab. This was done completely independent of what Kai does, but it led us to work together, actually, which is a nice story that happened here. Kai's also working on Codex now, and I'll show some of his data later. But it's really a testament to me that the system is reliable. It's incredibly sensitive, and it produces robust data. So this is a good example. I like to lead in my talk usually with this example on how this single-cell spatial phenotyping works. And all of this was done with the Codex technology. So I'm next going to give you a very brief overview of how the technology works, how the experiment is done. So the Codex system, you can see that here on the left-hand side, is essentially a microfluidic device. It's this blue and white box. And this box connects, you can see, via this tubing into an epifluorescence imaging system. So in this case, you see this gray and black machine. That's a Kien's BZX imager. So essentially, that's an automated epifluorescence microscope. And the connection between the two is this fluidic stage. So you can see the stage sitting in the middle of that Kien's is connected via tube into your Codex box. That's sort of your hardware and your mechanics around it. So Codex really is a microfluidic device. You can pair that with a wide range of epifluorescence or even confocal microscopes. So Kien's is probably the most commonly used at the time, but Zeiss axioscopes, we're seeing increased momentum with different builds right now. But that's a nice thing. It's essentially, it's designed to be plug and play with optical imagers. Again, we're relying on optical imaging. This is technology that's been around for many, many, many years and has been refined over and over again to give us the optimal resolution. Now, the chemistry of Codex is really, that's our product. This is where the magic happens. So if you look at these 3 circles here in red, blue, and yellow, let me just walk through this here. This circle is your tissue. And in the first step, what you do a one-step staining of your tissue. So that means you take all 40 or so antibodies that you have, you put them on your tissue all at once, incubate it, tissue stains. Now, each antibody is labeled with an oligo barcode, a unique oligo barcode. So type your antibody 1 will have oligo type 1. Your antibody number 2 will have oligo number 2. So that makes the antigen detection distinct from one another. Once your tissue is labeled, it actually goes on to that Codex stage. You close up your machine, and the system's automated from here on. So you can go home, go to sleep. Now, what happens while you're at sleep, or in my case, in the karaoke bar, the Codex system takes fluorescent reporters, and it does a cyclical imaging. So where if you now go from the red tissue state to the blue tissue state, it will first identify 3 antibodies. So what it does, it dispense 3 fluorescent reporters that are complementary to oligos on 3 antibodies. The microscope then takes a picture. You can see that little picture, cycle 1, underneath those tissue images. That will be a representative 3-color image from the first cycle. Once that picture is acquired, the Codex machine washes off the reporters. Tissue reverts back into its native state. That's the red circle here. And then 3 different reporters are put on. The microscope takes another picture. That's the yellow thing here. And then that will be cycle 2. And it repeats itself over and over again until you've essentially read out all of the antibodies that you labeled your tissue with. There's no stripping involved. This is an isothermal reaction. This is automated. And the reagents are all inventoried. So you can buy most of this stuff off the shelf. You don't have to mix stuff. You don't have to come up with recipes. It's extremely convenient. Right now, at the moment, if you were going to do this experiment, you have access to 42 barcodes, which means you can put on 42 antibodies with tendency increasing. So we're expanding our barcode menu because we realize people want to see more and more different outputs. So that's essentially how this works. It's a cyclical IHC where you label your tissue with inventoried reagents and essentially very clean. You don't have a lot of cross-reactivity because you're not using secondary antibodies. And that's a very, very nice thing about this. But I'm just going to come back one more time, full circle under this idea that this is a whole tissue imaging experiment, meaning that whatever tissue you can fit onto the cover slip that we use, you can image that in its entirety. It's up to you as a user what you're going to image, and you have single-cell resolution. And I'm just going to say a few words why we think that this is very, very important. So here again, we're returning to the same breast tissue that I've talked about before. This is just DAPI, but I love this image. I just think it's beautiful. You can see these different areas here of like hyperplasia or swollen regions. This is not a healthy looking tissue, but again, we're seeing every single cell. So if you're used to taking images DAPI, this is what you expect to see. It's a single cell resolution of a very large piece of tissue. Now, if you look around, a lot of the technologies that are available today, they actually depend on ROI-based imaging approaches. So rather than imaging a whole tissue, the systems, they need predefined grids of ROIs, or the users select certain ROIs because the technology only allows them to look at so much substrate at once. There are, of course, many reasons for that. This is by design, mechanical or analytical. This is just how some of these systems work. And what's really astounding about these approaches is that you actually end up losing a whole bunch of data. So let's just go back quickly. This here is a whole tissue breast, whole breast tissue. You can see there's a lot of biology going on. There's a lot of regionality. I mean, there's something very different, this particular region than, say, this packed region of epithelia. So there's so many different layers that you can slice this and see different things. But now let's go to an ROI-based imaging approach. You cannot really make out any of this regionality anymore. All you have is a bunch of ROIs with cells inside of them. And we did some modeling here. So we overlaid Codex data on just a grid that, for example, you would use in a spatial transcriptomics experiment. And we find that you lose up to 49% of your data with some of the original designs. Again, this is done by necessity or by the nature of the technology. But the fact is that you end up losing a fair bit of data. And if you think about it even further, a lot of the technologies today that we're using for spatial biology experiments don't actually have single cell resolution. So now you cut out 49% of your data because you do an ROI-based imaging approach. But what I've done now is I actually blurred each one of these circles by the diameter of how big these circles are. So assuming you have a technology where you're imaging 50 micron diameter ROIs, without single cell resolution, you end up really compressing your signals down, averaging your signals out a whole lot. So essentially what you see right now are fuzzy spots. Really, that's what it is. So the question at this point is what remains. Is this actually still single cell biology? In my mind, it is not. And I'm going to give you an example as to, I really like this example, but I'll give you an example that hopefully it illustrates to you very clearly that it's very, very important that we do indeed see every cell. So here I've juxtaposed this ROI-based approach versus a non-ROI-based approach to analyzing data. So on the left-hand side, you can see a signal for CD34. And what we're doing here, and this is just Codex data, just a single antibody channel. It's not cleaned up. You can see all kinds of fuzz here in these sort of bluish little tiny spots. But CD34 is a label for the vasculature. And vasculature, there's a long tube. So you can see these strings here on the left-hand side all over. They're like short strings and long strings, fat strings, thin strings. That's what the blood vessels look like, though that is your vasculature. So without knowing too much about, without even caring about the vasculature, frankly, you can see where it is. You know where the arteries are, you know where the capillaries are. You could probably see a broken vessel. Now, if you look at the right-hand side, these colored dots here are the intensity readouts for the very same signal. So that's your CD4 pattern right there. You can see a few orange spots. You can see some dark blue spots. So you probably can say, oh, there may be a blood vessel there, and there's certainly not a blood vessel in the dark blue spot. But you're missing a lot of granularity. If I was going to ask you, where do you think, the capillary is versus the arterial, I don't think this is really possible here. So I think, to me, I appreciate the importance of not, let's say, compressing your data down for the expense of gaining something else. I think it's really, really important that when we do these types of experiments, especially when they're single-cell biology experiments, that we maintain this information. And that is really what the power of this, not just a Codex experiment, but any spatial biology technology that is using optical microscopy to reveal these signals. So just to compare, once again, we're looking at the same tissue, about 4 by 2 millimeter breast tissue, 63,000 single cells were mapped with Codex. Now, if you break this down into ROIs with about 10 cells per ROI, you have about 1,400 different ROIs. So those are 14 points of analysis versus 63,000. It's quite a difference. Both of them are looking at the same thing, CD34. And the conclusion here really is, on the left-hand side, which is a Codex experiment, you're doing single-cell spatial biology. You could tell me where the blood vessels are. The right-hand side, I would call this a spatial approximation. You see, there's a regionalization, there's some blood vessels here and there, but that's about as much as you can say. And I think there are a lot of purposes for both of these technologies. They're probably more complementary than anything, but it's very important to have this distinction, especially when you're new to this field and you're planning to do this type of an experiment. What is it that you want? Do you want to do single-cell spatial phenotyping, or do you want to approximate the space? I think this is a question everyone needs to ask themselves in the very beginning. Both things are possible today. I think the end goal will always be to do single-cell biology. We all know this. The CZI stated goal is to study every single cell. So a choice is easy from here on. So let's move on to a little different topic then. So now I talked to you about what this experiment looks like, and kind of going back and forth with what data structure is. I'm going to just talk a little bit about what you can do with these data now, because I think there's probably a lot of you have worked with single-cell RNA-seq. I just want to point out some of the similarities in the data, and then give you sort of an idea where we think the field is going to be going soon. And the title for this section is Phenotype, Meet Your Phenospace. So I think this is the first time I've said this in public, phenospace, and that term's already been used for a completely different type of research. But what I'm trying to get to here is that we all talk about single-cell phenotyping today, but one of the things that we at Akoya are really starting to appreciate is this idea of contextual cell phenotypes. A phenotype is, sure, it's the idea of a cell, but what if you put that cell somewhere else? Like, imagine yourself, being at home on your couch. Are you the same person who's sitting in his or her office, wearing, professional attire or so? We think the same thing happens to a cell. Whether or not the cell is sitting at home on its couch or if it's gone into the wrong place, like a tumor, the cell would kind of have the same phenotype, but is it going to be behaving the same way? And so that's what we think contextual phenotype would capture. And really, probably this is the next frontier in my mind where single cell biology should and will go. So this is an experiment that we did together with Kai. Again, he's at UCI and he's a CZI investigator. This is one of the first experiments that he did. We did this last year. And once again, this is breast tissue, and it's just gorgeous, this tissue. So what we did in this experiment, we labeled the tissue with 32 antibodies. There's on this particular image, there are 81,000 cells. It's pretty large, it's 6 by 8 millimeters. And you can see sort of these round things here. These are one type of epithelium, a secretory epithelium. That's where the fluids are generated. And then you have these long lines. These are actually tubes. And so that's where the fluids will be collected. And you'll notice there's a whole bunch of black space around all this. That's actually all fat and connective tissue. We didn't put any markers for fat or connective tissue labels on there, but I love this image because it illustrates the luxury that we have at Codex. Think about having an ROI-based imaging approach. Like, would you waste your ROIs on fat tissue? We don't care because we can just image the whole tissue. It takes a little bit more time, but now, of course, Kai's asking, of course, now, what's in the fat? So he's getting interested in that. But having access to whole tissue imaging, it's a luxury, but again, it's necessary to understand what's going on. So what we do with this breast tissue is label it with 32 markers, segmented out each cell, and then we break it down so we can get heat maps for every single marker in this experiment. And so you can see BCL2 is expressing in red. So this is your top left marker here in the very top. You can see when it's red, there's a lot of expression for it, and blue, it's cold. If you go, so for example, to the very bottom left, there's TP63. That's a marker for basal cells, predictably expressed throughout the epithelium. Sometimes you get markers that don't really do what they're supposed to. Like this is immunohistochemistry. You will always get non-specific binding, and so these types of analyses really help you pick this out. So it's a fantastic way to not only represent your data, but to do QC and see what's going on. And then on top left, you can see a UMAP of your clustered data. All of this is done in an end-to-end analysis solution. And it allows us to, very quickly quantify your different cell types. So you can see we quantified about a dozen different cell types here. We have a lot of luminal cells, type of epithelium. That's that orange pie square. You have basal cells, a smaller population of cells, and so on and so forth. All of these cells were expected to be there. Codex experiment works really, really nice for breast tissue. So once you have all your cells segmented and phenotyped and annotated, this is really where the exciting stuff begins. So this is where, I say, phenotype meet your phenospace. So here you can see about 10 different phenotypes, CD4 cells, CD8 cells, dendritic cells, mapped out on top of the actual architecture of this tissue. So each one of the cells has now become a dot with phenotype. That's what it looks like. And if you look a little bit closer, you can see these dots a little bit better. So we've become really interested in this idea of mapping the connectivity between every single cell in your image. And that's what these lines here demonstrate. So these are triangulations, essentially just trying to figure out what the shortest distance between any three neighbors are. When you do this type of an experiment, essentially you can draw a line from every single cell to every single other cell on your tissue. You're just generating a lot of data. So it becomes very interesting or very complicated trying to understand what's going on here. And one of the concepts that we've been excited about and that's sort of come out in recent months has been this idea of neighborhood analyses. So I'm going to walk you through one of these. So assuming we're interested in CD4 T cells, you can see that on sort of in the middle column in these little cartoons here. There's a CD4 T cells are red in this image, and there's one T cell in the middle in each one of these images. What we had the computer do is we interrogated the entire breast tissue, and we said, now take a look at every CD4 T cell and break it up into different neighborhoods. What do the surroundings of these T cells look like? And when you do this, the computer comes up with different patterns. And it gives you sort of an idea that there are certain parts of the tissue where a T cell is more likely to be next to other T cells, as you can see in these series of cartoons, than in other parts of the tissue. So I just made another cartoon here. Now you can see these little T cells. They're no longer surrounded by, the same type of cell, but instead they're actually surrounded by epithelial basal cells. So you can see the T cell in red, and there's all kinds of yellow cells around it. So now what we've done, this is just the way the tissue is. We're just taking an objective look at it, and it's sort of a clustering-based algorithm. We're looking throughout the entire tissue, trying to figure out where the different cells would be here. And I just wonder when I look at this, I'm like, what does CD4 T cells in cluster 1, how would they behave compared to the CD4 T cells in cluster 6? Or if we just take this analysis further and we look, for example, at luminal cells, you can see 9 different clusters for these luminal cells. The luminal cell here will be the brown cell. Each one of these rows shows you a different environment in which a luminal cell can exist. So look at cluster 1. You have a little brown cell surrounded by CD4 T cells always. Let's look at cluster number 4. You have luminal cells that are somehow associated with lymphatic vessels. Cluster number 7, luminal cells next to dendritic cells. You can go on and on. Now, these are all luminal cells by phenotype, but every one of these phenotypes, every one of these cells has a different neighborhood. So this is, I think, a concept that is really emerging now, and Gary Nolan grandfathered this. This came out at the end of last year, this idea of cellular neighborhoods. And what they've shown in that paper, and I think it's a really great paper, I encourage anyone who's interested in this to read through this first and then go from there, is that these types of patterns matter. So they looked at colorectal cancer samples with 2 different types of colorectal cancer with different outcomes, and they show really nice correlations between certain types of neighborhood being present in the tumor and stromal areas and the actual outcome of what these diseases are. So when the phenotypes meet the phenospace, the cell's behavior probably changes. And these are things that we need to be aware of because they seem to actually have clinical manifestations. They seem to matter in the larger scale, in the larger picture. So we're hyper aware of this right now. And of course, by having single cell whole tissue resolution, we are very much able to actually interrogate these types of phenomena. And it's very new, I think not so new anymore, but this is a more and more growing field. And I'm very excited that we're working on this. And you'll probably hear a lot more about this going forward. So this was sort of a bit of a deeper dive into Codex data. And now I'm just going to sort of conclude my section with a few case studies. I'm going to try to hit a few frequent questions that I get about what can you actually use Codex for? And I'll just give you the answer right away. Really, any tissue you can image, in theory, any tissue that you can image and do IHC on, you should be able to do Codex on because it is IHC, so immunohistochemistry, I mean. And let's just walk through a few of these. So recent months, we spent a lot of time actually working on brain tissues. So this is a collaboration with Dr. Bahare Ajami at the Oregon Health Science Institute. Very interesting tissues that we have here. But what you can see here is a piece of human brain tissue, formalin fixed, paraffin embedded, highly autofluorescent tissues. Most people think you cannot really do clean imaging on these tissues, but there are ways to do that with Codex. It revolves autofluorescence removal and then subsequent imaging. But as you can see here, it's just gorgeous staining. So in blue, you can see amyloid plaques. This is sort of a pathological manifestation of Alzheimer's disease. And in red, you can see glial cells. These glial cells are like surrounding these plaques. And then in green, there are nuclei. Now, of course, you can label the single tissue with 42 different antibodies. So you can start to interrogate the immune microenvironment of Alzheimer's really nicely. So you can see in these little images that I just popped up here, each time it's EBA1, So EBA1 shown in red is actually what's called microglia. So I guess for those of you who are not neuroscientists, think of a microglia as something like a macrophage of the brain, and they have different activation states. So on the left-hand side, you can see EBA1 that's co-labeled with TMEM119. So these will be presumably active as they're associated with this blue big protein plaque. So there must be a lot of action here. If you look over on the right-hand side, you have the same cells labeled with different immune markers, once again showing activation states. Really nice to see this work together. And now if you have any 4D markers you wish to put on here and study these tissues in more detail, it's possible to do so. So we're very excited about that. Very common question that we always get is, can you actually image tissue microarrays? Yeah, the answer is yes, you may. It's the same really as for a larger tissue. You just need to be able to fit them onto the cover slip. So you can see here, this is a tissue microarray that we got from Dr. Patrick Micke. He's up in Uppsala. And these are all lung adenocarcinomas. I think the entire microarray that we imaged at the time had 50 cores or so on it. And I'm just showing a fraction of these. But again, these were labeled with Codex with, 24 antibodies or so. Now suddenly you have a large sample size in addition to all of these biomarkers. This is really cool. Like you have all of these individual patient tissues with the same setup, 24 or 30 different biomarkers. Imagine how much data is contained within that one single experiment. It's a lot. Like I spent, weeks just sitting over these data and seeing things. And so it's really exciting. We've done work in neuronal cultures and organoids. So the nice thing here is, especially with cell cultures, you can grow them up right on our cover slip. That's what we've done with Dr. Schule at Stanford University. So they cultured neurons right on top of our cover slip. And then we took that cover slip, we fixed the neurons, and we ran a Codex experiment with it. And the neurons don't peel off, they don't fall off, they just stay there. You can see that right here. So this is a snapshot from one of these cultures. In green, you have glial cell. Yellow, you have neurons. And then in cyan, you can sort of see these stringy patterns here. This is certain markers for neuronal filaments. So really nice tissues, really nice versatility of the system. And my summary slide then would just be this, can I use Codex for really any tissue that can be imaged. So just this last year in our lab, we've seen melanoma, brain tissue, lung tissue, skin, gut, kidney, even blood cells we've imaged by fixing them on a cover slip. The breast we talked a lot about, and spleen. I should add here that this works, of course, for mice and for human tissues. It works for fresh, frozen, and FFPE. Really anything that people work on nowadays for IHC seems to work quite well with this system. So it shouldn't be a question of what, it's just, when. So with that, I'm going to conclude my section of the talk. I'd like to thank you for your attention. And if you want to hear more about Codex or our Phenotype system, which I didn't have time to talk to you about today, visit our website at akoyabio.com. And I'm looking forward to the Q&A now, and I think there's a lot of interesting questions pouring in. So I'll hand the mic over again.
Thank you, Oliver and Jonah, both for your excellent presentations. And yes, it is now time for our live Q&A portion of the webinar. As a reminder to our audience, if you have any questions you would like to ask, please do so now. Just click on that Ask a Question box, type in your questions, and click Send. We do have a lot of great questions that have already come in, so let's get started. Let's go with this one. Let's start with Andrew's question. A very cool presentation. It sounds like your team at Akoya performed Codex imaging and Codex data analysis for Kai. Is this something Akoya is offering to other investigators or industry partners?
I guess I will take that question here. So Kai is a collaborator of ours. He works together with us. And we have many collaborations, of course, with both academic investigators and industry partners. We have a technology access program for Codex. It's called STEP. where we do both experiments in the wet lab and dry lab analysis with really anyone. So it's possible to get our attention for sure for collaborative purposes. The best way to do that is to come in through our Tech Access program, which is called STEP. So you can inquire that to any one of our reps.
Thank you, Oliver. Jonah, let's go to you for this next question. Most of the single-cell funding opportunities posted by CZI are limited to normal tissues. When will CZI be announcing new opportunities for diseased tissue?
Yeah, that's a great question, and that is absolutely right. So historically, within the single-cell program, the vast majority of our RFAs and grant funding has gone to normal tissue. I would go back and highlight maybe two things, one that I spoke about, one that I did not. First was the inflammation program, which was launched last year. Those are relatively short initial projects, so 2 years in duration. And it's really our hope and ambition that those will grow and lead to additional funding opportunities. So, we don't have any of those active right now, but certainly hope to see our work and support of that area grow in time. The other that I would highlight is the Neurodegeneration Challenge Network opportunities. And there are a number of groups using single-cell methodologies. Many of them are very, very interested in spatial organization, and you can imagine the importance of these sorts of questions, for example, in relation to the immune system and its interactions with plaques, for example. So that is also another program that I didn't touch on that is CCI related. And in both cases, we would also encourage people to reach out with disease data if it's interesting to them to use cell ID. So again, that platform is available both to our grantees, but also to anyone else. And for example, a large number of groups have used or contributed data to help disseminate and make work on COVID go more accessible. So that's one area where we're absolutely supporting disease work, and I say that there will be many more funding opportunities for that and neurodegeneration program. Great question.
Great, thank you, Jonah. Okay, for our next question, it's two parts. To what extent can the phenotyping panels be customized? For example, if I wanted to differentiate cell types within kidney tissue, and I know the markers I'd like to test for, would it be possible to generate an assay that I could use to test tissue from, say, a wild-type mouse versus a mouse with a genetic modification?
Yeah, sure. I'm happy to answer that question. So the phenotyping panels actually at the moment are fully customizable. We have inventoried antibodies that you can choose from in a catalog. So there's many antibodies that are already pre-configured to be used for Codex. A lot of immune markers, a lot of structural markers, oncogenes, and it depends really what you're interested in. So you can buy those off the shelf. But really the customization that will be then of interest to you is all of the antibodies that you already are interested in, that in some cases you have in the lab. You can actually chemically modify them, that is, conjugate them to our barcodes. So let's say you're working on 24 antibodies that do not exist yet in a Codex inventory. That's not a problem at all. That just means that you buy the conjugation kit, you perform the conjugation yourself, or you get it in through our technology access program where the conjugation can be done, and you build your entirely custom panel. So every reagent that you need to generate a fully custom panel is available to you. There really aren't any limitations there. So yeah, you can, anything you want. You can look at kidney in mouse, you can look at kidney in human, disease, normal. You have to make the design and you're good to go.
Thanks, Oliver. And a quick question again for you. Do you track single cells?
Yeah, that's what I just said for the last half hour. That's all we do. That's our bread and butter. Everything is single cell.
Very good. Thank you for the clarification. Our next question. Is this frozen section or a FPPE tissue section?
The large majority of the tissue that I showed today were FFPE tissue sections in human, except for the cell culture image at the very end that wasn't even frozen, that was live fixed. So FFPE works really well.
Thank you. How long does it take for a full panel of 40 antibodies to run through? And will you be covering data analysis in this presentation?
That's a great question. So the 40 antibodies, it takes a fair bit of time, but it really depends on how big your tissue is. So if you're only looking at, a small, 400 by 400 micron tissue, you probably run it through in less than a day. But if we have a lot of people who are running really large pieces of tissue, let's say 10 by 10 millimeters on 40 antibodies, it can almost take 2 days, or 36 hours. So it's a factor of how large your tissue is, because the imaging happens in tiling manner. It's a sort of a serial process, plus how many antibodies you have. More antibodies you have, the larger your tissue is, the longer it takes. And with regards to the second part of your question about covering the analysis, I did not cover how the analysis is done. I've shown some example of what can be done. We have a lot of webinars on our website on how analysis can be done. I encourage you to look at those if you're interested. We did quite a few sort of tech talks in the last few years, and there's a ton of information in the literature and webinar section of our website.
Thank you. Let's go with this next question. Is microbiome included in the Human Cell Atlas?
Yeah, I can take that one. So it's an interesting question. The short answer is it's certainly not at the core, but for many tissue types, and especially barrier tissues, there is, of course, an appreciation that the microbiome is a key factor in those neighborhoods and the cellular interactions that Oliver touched on and had some beautiful images for. So I think the answer is that it's not necessarily the core, but certainly within the CZI-funded groups that are working on developing and contributing data to the Human Cell Atlas, it is certainly an assay that some of those, an assay and data type that some of those groups are collecting and measuring. So for example, certainly within the airway and gut and places like this. So it's a little bit of a wishy-washy answer, but in some cases it is, and we would hope that all of that data would be associated and linked, but certainly not true of all of the organs and tissue types.
Thanks, Jonah. Our next question. Is phenotyping solely based on thresholding of biomarkers signal intensities?
At the moment, phenotyping is solely based on thresholding, not always thresholding, but cataloging biomarker intensity. Yes, it's at the moment that's the main method, but we're exploring other ways of doing it, because there's quite a few different aspects we're finding, especially with cancer cells, and nuclei have very different shapes in immune cells. So the main tool, and it's always been like that, and it's consistent with other platforms, is to record and threshold biomarker intensity. But we're finding more and more, since we have the resolution, and we can see, we can see the shapes of nuclei, the sizes, geometry. We're exploring ways to add those factors on top, because it gives us a much more granular understanding of the phenotypes, and the results are much cleaner. So I would say that's actually a rapidly developing space.
Thank you. Now we have time for a few more questions, so let's squeeze some in there. How about, can this technology allow quantitative comparison between disease versus normal?
It should, absolutely. Any comparison can be made. So we've done a lot of this work with our Phenoptics platform. That's a standard experiment there. And with Codex, I've seen more and more people are doing a lot of quantification of phenotypes. So, for example, there are more T cells in one type of cancer than another. So these comparisons are absolutely possible. And it's becoming routine, actually, to start working on these.
Thank you. And while we're speaking of phenotypes, let's go with this question. How do you see Codex and other types of multiplex spatial data becoming standardized across modalities and platforms around shared definitions of cell phenotypes and phenospaces?
I think that's a great question. I don't exactly know how it's going to happen, but I'm an advocate for that. It must happen. So Codex is the new kid on the block here. It's only been around for 2 years or so. But already there's an interest from several big groups to achieve parity and standardization across sites. For example, we know that the Human Tumor Atlas Network is quite actively pursuing these types of standardizations for cross-platform. So work is being done right now actively across the spectrum to try and see. First question, obviously, is what is parity? How do we define it? When can we get it? And then the standardization is probably the next one. I should also add that other technologies such as multiplex immunofluorescence, which we call Phenoptics at Akoya, is already reached a level of standardization that's very good. So it's happening, and I hope that we come to consensus in the community very soon. It really depends on the community at large to drive this. We have, of course, at Akoya, have a big responsibility to push this forward, and we take this issue very serious.
I would chime in on that one and echo Oliver's comments as well. This is something that's really important to us, and also have enjoyed and appreciated the commitment and excitement about this that the Akoya team has. And so I think it is very much a community-based one, and will take collaboration and openness among the groups generating the data and the companies that are enabling that, as well as opportunities and willingness to share data in interoperable formats and with common panels and things like this. But really, really important problem that we all will have to work on in the coming years.
Great. Thank you both. And we have time for one last question. We'll wrap with this one. What was the objective you used for many of your images?
Most of the images you saw today was just done with a standard 20x lens. The reason for using a 20x is that we like the whole tissue imaging approach. So when you're imaging a 10 millimeter by 10 millimeter tissue, it's just more economical to work with a 20x, and we get what we want out of a 10x. So what you saw today was 20x non-immersion. You can work with 40. Actually, obviously, the resolution is even better. The granularity is better. The phenotyping is cleaner. It just takes much longer time to map these big tissue parts. So we have both lenses in regular use. Depends on what your question is. Some people use Codex to look at a restricted area. Say, for example, they want to look at transcription factors or even nowadays, like synaptic terminals. That's obviously a 40x workflow. But everything I've shown you today is that it is large tissue, whole tissue analysis is easily done on a 20x.
Thank you both for this excellent Q&A session. I do want to remind our audience that there were quite a few questions that we did not have time to answer today. Those questions will be answered via email to the email address that you provided at the time of your registration. I also want to mention that this presentation is available for unlimited on-demand viewing. We will also be adding a link into our on-demand studio that will allow you to reach out and provide any feedback, UX insights on cell by gene. So please come back for the on-demand to view that link. I want to thank Dr. Cool and Dr. Braubach one more time for their excellent presentations today and the very informative Q&A session. If you missed any portion of today's webinar, just a reminder, you can come back and watch it on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. And then as a reminder, today's presentation is educational and it does offer continuing education credits. Please click on that continuing education credits tab and follow the process to obtain your credits. Thank you so much for joining us today. Have a great day.